When we consider the morality of cloning, one initial hurdle is to get past all of the misconceptions that get peddled in the media and in movies. We won't have a solid grasp of the ethics of cloning if we misunderstand what cloning is. This is why the excerpt from Dina Davis's book, Genetic Dilemmas, is extremely helpful for us. She describes in detail what is actually involved in cloning. She begins by noting that there are two kinds of cloning: blastomere splitting and somatic cell nuclear transfer, which can be abbreviated as SCNT. The first, perhaps best considered as quasi-cloning, since it doesn't result in a new organism from an adult, happens when an existing embryo is split into 2, 4, or 8 new embryos. In early embryos, the cells are what is called totipotent, which means that each cell has the potential to develop into an entire new organism. Thus, when the early embryo is split into 2, we would have 2 independent organisms, which would, in principle, both develop into fetuses, newborns, toddlers, and so on. As we can see, this is a process of an existing embryo being split into several other existing embryos. The second kind of cloning, the kind that most people think of when the word cloning is mentioned, is called somatic cell nuclear transfer. There are two kinds of cells in the body: germ cells and somatic cells. Germ cells, such as the sperm and egg, have half the number of the organism's chromosomes. This is because the zygote that results from the fusion of the sperm and egg itself should have the normal number of chromosomes. If the germ cells had the normal number, then the zygote embryo would have twice the normal number. Somatic cells, that is, the cells of the body that aren't involved in reproduction, have the normal number. The genetic material in somatic cells is half from the organism's father and half from the mother. With SCNT, somatic cells are used for reproduction. A healthy cell is scooped out, which removes almost all the genetic material, and a somatic cell from a donor is placed into the now empty egg cell. This egg cell, with a complete set of genetic material, will now develop into an embryo. Notice, however, that the complete set of genetic material comes from one donor, rather than two, as in sexual reproduction. Normally, half of the genetic material would already be in the egg cell, and the other half would come from the sperm cell. In the new case, the genetic material of the egg cell has been removed, other than a trace amount that remains in the wall of the cell. There is no sperm cell involved, so there is no genetic material present except that of the donor. The donor doesn't have to be the person to whom the egg cell belongs. Thus, in this case, the parent of the child that is born won't, strictly speaking, be the person who gives birth to the child. It will be the donor. More than this, the parent will also be the identical twin of the child, what is called the delayed or asynchronous identical twin. Stranger still, since the donor is considered the parent in some sense, the cloned individual will have a parent as a sibling, and since the genetic parent of the donor is his or her parents, the cloned individual will have grandparents who are his or her genetic parents. It gets very complicated, so perhaps we should distinguish several senses of parent. First, we might talk about a biological parent, defined as the person who gives birth to the child. Second, we might talk about a genetic parent, defined as a person half of whose DNA gets passed down. And third, we might talk about a clone parent, defined as the person who donates a somatic cell for the purpose of cloning. Now imagine Denise, a child who is a product of cloning. The cloning process occurred as a result of an egg cell of Mary's being scooped out and replaced by a somatic cell donated by Jane. Mary's egg cell developed as fertilized egg cells do, and after a pregnancy lasting 9 months, Mary gave birth to Denise. So far we have Mary as Denise's biological parent, since Mary gave birth to Denise. Jane is Denise's clone parent, since Joan donated the somatic cell. Jane and Denise are also identical twins, since this is defined as having identical genetic material. Jane's parents, John and Ruth, are Denise's genetic parents, since half of John's genetic material and half of Ruth's genetic material make up for the complete set of genetic material of Denise. Insofar as Jane is a parent of Denise, a clone parent, John and Ruth would also be grandparents to Denise, clone grandparents. To make matters more complicated still, since Mary is Denise's biological parent, her parents would be Denise's biological grandparents, even though their genetic material is almost non-existent in Denise. We can see from the full description in Davis's book that cloning is just another form of reproduction. It's not, as some movies are made out, anything like Frankenstein's creation of a fully formed organism or a kind of Xeroxing of individuals. If you wanted to create a clone of yourself to take exams, for example, so that you could sleep in, first, you'd have to wait 18 years or so for the clone to be the right age. And second, the knowledge you gained by attending class would not belong to the cloned organism, any more than, if you had an identical twin, you'd be able to draw from what he or she learned by going to class. Clones are autonomous, independent individuals who share an identical set of genetic material with another person who is of a different age. Once we understand what cloning is, at its fundamental level, we can survey some of the arguments made in favor of or against it, based on moral reasons. We can also see whether cloning for different reasons or purposes affects its moral status. Davis, in the excerpt from her book, argues that moral considerations about cloning depend crucially on the motivations of persons pursuing the possibility of cloning. Michael Tooley, in the article we'll discuss in class, considers two contexts in which cloning might be used. A, cloning for the purpose of harvesting organs, and B, cloning fully autonomous persons. He considers arguments against the morality of A and finds them all unpersuasive. Regarding B, Tooley concludes that the present state of scientific knowledge about cloning mandates that human cloning not be attempted at the present time. It stands to cause emotional and physical suffering both to the parents and the persons that would develop from the cloned cells. In principle, however, Tooley finds no moral reasons against cloning persons. Indeed, he finds several benefits to cloning, which we'll discuss in class. And Leon Kass famously claims that we all feel a kind of repugnance, and rightly so, when we hear about cloning, and there is deep wisdom in this repugnance. Part of how we find meaning in life is bound up in the act of sexual reproduction. Moving toward asexual reproduction, Kass argues, stands to demean our own existence. We'll talk about all of these issues concerning cloning in detail in class.